Hello, my name is Radit Ephraim, and I will be presenting to you about antibiotic-producing microbes found in soil in collaboration with Tiny Earth Project Initiative, TEPI, and Dr. Laurie Scott. The antibiotic crisis is a growing concern in the United States as more and more diseases are becoming untreatable due to antibiotic resistance. According to the CDC, 23,000 people die as a direct result of contracting an antibiotic-resistant infection. Many of these infections are caused by nosocomial pathogens or infections that are picked up in hospital and are likely antibiotic resistant. Antibiotics are secondary metabolites of bacteria that inhibit the growth of or kill other microbes. Bacteria are prokaryotic microorganisms that reproduce by binary fission, a process by which every parent cell yields two daughter cells. Of all the bacterial strains, the ESKAPE strains are the most dangerous, and you can see those listed here. There are 6 of them. They are dangerous because they are multidrug resistant, and there are 5 strategies described to combat the ESKAPE strains, as you can see here. Antibiotics in combination, bacteriophage therapy, antimicrobial peptides in therapy, photodynamic light therapy, and lastly, silver nanoparticles in therapy. The point of study in this research, however, will focus on testing soil samples to see if soil microbes are capable of producing antibiotics. In this study, ESKAPE-like strains, Bacillus subtilis and E. coli will be used. They are model organisms for ESKAPE strains as they are similar and provide comparable data in a safe manner. In this first figure, you will see 2 streak plates and then 2 spread plates that have been labeled 1 through 4. In the first plate, you will see a streak plate of Bacillus subtilis. Large colonies are visible, indicating a high growth rate. In the second is a streak plate of Erwinia cerativora. Visible colonies are much smaller, indicating a slower growth rate. And the third is a spread plate of Bacillus subtilis with unknowns A through D patched onto them. Around A, you observe a halo, or zone of inhibition. This is something we are hoping to observe with our soil isolates, as a halo indicates that unknown microbe is secreting antibiotic, effectively inhibiting the growth of Bacillus subtilis around it. In the fourth plate, we see a spread plate of Erwinia cerativora with unknowns A through D patched on. There are no halos observed. All of these plates were incubated at 28 degrees Celsius for 24 hours. In an effort to combat antibiotic resistance, new sources of antibiotics are being sought after. This project is part of a nationwide search for new antibiotic-producing bacteria in soil. TEPI aims to educate the public about the antibiotic crisis and discover new antibiotics with the help of students conducting research such as this. The purpose of this project, then, is to collaborate with TEPI and identify microbes in local soil that have antibiotic-producing abilities. Now moving on to materials and methods. Unless described otherwise, the bacterial strains and protocols used in this study were provided by the Tiny Earth Project Initiative. We started with collecting the soil sample. We made sure to do this in a sterile environment to make sure that the only bacteria we collect is from the soil and not our hands or shoes. Next, we continued to make serial dilutions in an effort to avoid a plethora of bacterial growth on the plates. The latter 3 dilutions were used in order to make 9 plates on LB, 10% TSA, and PDA. Colonies grew, and from those, the largest and most diverse were used to create a set of master plates. These plates are the plates that we will refer to for further testing and the creation of other plates. Using the master plates, we created a set of tester plates with Bacillus subtilis and E. coli to see if any of them created any halos or zones of inhibition. The microbes that did create a halo were then streaked onto a plate and then retested to confirm that they are in fact antibiotic producers. From there, a polymerase chain reaction was done to amplify and copy the DNA, and gel electrophoresis followed. The purified DNA samples were sent to the Iowa Institute of Human Genetics, and while we waited for the DNA sequencing results, biochemical testing was done to help narrow down the identity of the isolates. When sequencing results came back, they were put into BLAST, which generated possible genera for the strains. It's important to note that the soil sample was taken from a yard in Rock Island, Illinois. Samples were incubated at 28 degrees Celsius, apart from biochemical test plates, which were incubated at 35 to 37 degrees Celsius, and the forward and reverse primers chosen are the 27F and the 1492R, respectively. The results obtained here were pretty conclusive. In this first figure, we see the final streak plates of the top 4 antibiotic producers. In the first plate, we see isolate number 2, in the second, we see number 3, in the third, we see number 6, and in the last one, we see isolate number 15. Important things to note are that isolate number 6 is on a TSA plate, while the rest are on LB plates. And isolate number 15 has a strange yellow paint color, which indicates that it is probably not Bacillus or Streptomyces as previously assumed. In this next figure, we see bacterial strains plated on 3 different media: LB, 10% TSA, and PDA, using technique 2, which is a pitch and pack. The first column is an E. coli, while the second is Bacillus subtilis. We are looking for halos on these plates, and a clear halo is visible around number 15, as shown by the red arrow here. A halo is also slightly visible on number 6, as also indicated by that arrow. This was later confirmed in further testing. And the PDA plates showed very minimal growth. All plates were incubated for 24 hours at 28 degrees Celsius, apart from the TSA and PDA plates, which were incubated for an additional 24 hours in hopes of further growth. After the isolates were confirmed antibiotic producers, they underwent PCR to amplify the DNA, and then a gel electrophoresis was run. Here we can see wells 3, 5, 7, and 9 loaded with samples numbered 2, 3, 6, and 15 respectively. In the first lane is the molecular weight marker. We can see that the DNA are about 1.5 kilobases in length, which is what is expected from bacteria. The gel was run on 1% agarose gel at 100 volts for 30 minutes. Lastly, after the DNA results came back from the University of Iowa, I then input the data into BLAST and was able to identify my 2 strains. According to the BLAST analysis, isolate number 6, which is shown on top, is from the genus Bacillus, while isolate number 15, is shown on the bottom, is from the genus Stenotrophomonas. Some important characteristics to note are the query cover, e-value, and percent identity. Query cover is the percent that the sequence entered overlaps with the sequence in the database. E-value are the odds that a genus was a hit by chance. We want this number to be close to 0 if not 0, and here we can see that they're all 0, which is a good sign. The percent identity is the percent match between the sequence entered and the sequence in the database. We want this number to be as high as possible. As Bacillus and Streptomyces are the most common soil microbes, I conducted this research with the assumption that both strains pulled out would be one of these. However, isolate number 15 ended up being from the genus Stenotrophomonas, which is also a common microbe found in the rhizosphere of soil. Its purpose is to promote plant growth by secreting large amounts of antibiotics to kill anti-plant microbes. Information that is still unknown is how much antibiotic is produced and how much production is enough to inhibit microbial growth. The next step in research would be to determine the minimum inhibition concentration, or MIC, by serial dilution. Diluting the extract by a twofold dilution might be enough to determine the MIC successfully. This research was successful as antibiotic-producing bacteria were found and confirmed in soil. We are hopeful that the samples sent to TEPI for further analysis will make a great stride towards combating the antibiotic crisis. Thank you again for listening to this presentation on antibiotic-producing microbes found in soil.